Okay, now we're going to go over Chapter 8. This is an introduction to microbial metabolism. All right, so what exactly is metabolism? Well, metabolism is going to be the sum total of all the chemical reactions happening within a cell. And there's two types of chemical reactions, okay? There's catabolic reactions and anabolic reactions. You have catabolic reactions are going to break large molecules down into smaller molecules, and this releases energy. Anabolic reactions are going to be the exact opposite. You're going to make large molecules out of smaller molecules, and it requires energy. All right, you can't talk about metabolism without talking about enzymes. So enzymes are biological catalysts, and a catalyst is anything that speeds up a reaction. So enzymes are catalysts that are going to increase the rate of a chemical reaction by lowering how much energy it takes to start that reaction. Important facts about enzymes: they are not going to be altered in the reaction, permanently at least, so they can be used over and over again. And they're going to promote the reaction because they serve as a physical site for substrate molecules to interact. And I'll show you exactly how. It's called the induced fit model, and it's the way that they speed up reactions. All right, so we'll talk about this graph for just a second. Here on the x-axis, you have the progress of the reaction, the chemical reaction. On the y-axis, you have the energy state of the reaction. So here are our reactants right before the reaction occurs. This is our initial state. And then down here is the final state consisting of the products, and this happens after the reaction occurs. So you can see this line here is the reaction without the use of enzymes. So you have to put energy into the reaction to get it to go. That's called the energy of activation. And so you can see here you have to put more energy in to get this, this reaction to go without the enzymes than you do with the enzymes. There's a much smaller amplitude here. So enzymes bring down the energy of activation, or how much energy is required to make a reaction go. So what are enzymes made of? Well, first and foremost, they're going to be made up of protein. So some enzymes, simple enzymes, stop right there, and the entire enzyme is made up of a protein. However, most enzymes are conjugated or holoenzymes. What that means is that they contain a protein portion and a non-protein portion. The protein portion is called the apoenzyme. It is not active on its own. It needs a cofactor to activate it. And the cofactor is going to be the non-protein portion. For cofactors, you can have inorganic elements, things like iron, copper, magnesium, zinc, things like that. Or you can have organic molecules, such as vitamins. And when they're organic molecules, we call them coenzymes. So here's an example of 3 different enzymes here. You have the purple portion being the apoenzyme, right? The protein portion of the enzyme. And in this first one, we have a metallic cofactor. In the second one, along with the apoenzyme, we have a coenzyme. And in the third one here, we have a coenzyme and a metallic cofactor. This chart just gives some example enzymes and what their functions are. For example, catalase. I want you to be familiar with that one in this course because it's one of the biochemical tests that we do on the microorganisms. And what that does is it breaks down hydrogen peroxide into water and oxygen. And the cofactor required there is going to be iron. Other examples here, you have DNA polymerase. This synthesizes DNA. It catalyzes the synthesis of DNA, I should say. Cofactors required are zinc and magnesium. And then you've got a couple dehydrogenase enzymes down here that require vitamins as the coenzymes. Okay, so these enzymes are going to show primary, secondary, and tertiary protein organization, and I'll show you what that looks like in a second. But really what that means is that as the protein is being built, there are interactions between the atoms of the protein that causes it to fold in very unique ways that are specific to a certain protein. And that folding structure is what gives every protein its function. Some larger enzymes are also going to show quaternary structure. And again, I'll show you what that means in just a second. The active site, this is going to be the place on the enzyme where the substrate binds. Okay, and it's a very specifically shaped area that only very specifically shaped molecules can bind to. You can see here is the enzyme. Here's the active site of the enzyme. Here's the substrate, and you can see how the substrate fits into the enzyme, kind of like a lock and key. All right, so here we're going to talk about primary, secondary, tertiary, and quaternary structure of proteins. The primary structure is just the sequence of amino acids that make up the protein. Okay, it's just which amino acid comes next, essentially. The secondary structure, so as these amino acids are being added to the protein chain, they have these variable groups hanging off of them called R groups, and atoms of those R groups tend to interact with atoms of other R groups, causing the protein to bend. And it tends to bend in predictable ways. So it can form an alpha helix, it can form a beta pleated sheet, and not shown here, it can also form a random coil. Next, you have tertiary structure. So essentially what happens is these alpha helices and these pleated sheets, as they are bending, they're going to interact with other pleated sheets and other alpha helices to cause the protein to fold even more. So here you can see the protein appears to be folded in kind of a random way. But if you zoom into one of those folds, you'll see it's made up of even more folds. The beta pleated sheet here, or the coil here, the alpha helix. So that's what tertiary structure is. Think about it as superfolding, okay? Folds upon folds. Finally, you have quaternary structure. So most enzymes, they stop at the tertiary structure and they're functional, they're ready to go. Some enzymes need subunits, 2 or more subunits, each with their own tertiary structure, and they will come together and join and form a quaternary structure. Hemoglobin is an example of a protein with a quaternary structure. Okay, so how exactly does the enzyme speed up reactions? Well, it's called the induced fit model, and I'll just walk you through it here. So here you have the enzyme lactase, and it binds lactose. So lactose is going to fit into lactase just so, right? Because lactase has very specific active sites for lactose to fit into. Now, when lactose binds to the active site on the enzyme is going to change shape just a little bit. And we call this a conformational change. And when the enzyme changes its shape, it puts pressure or stress on these bonds in lactose, and it breaks them. And so you can see because it's putting stress on these bonds, it's going to require less energy to break the bonds. Once broken, the products are released, and the enzyme goes back to its initial shape or conformation. Now, this is an example of a decomposition reaction, right? We're breaking a big molecule down into smaller molecules. This is also called a catabolic reaction, right? You can do the exact same thing, just kind of the opposite way, with synthesis reactions or anabolic reactions, where you build a larger molecule out of smaller molecules. The molecules will fit into the active site of the enzyme. The enzyme will change shape, and that will bring the molecules closer together so that it's easier to form a bond between them. The big takeaway from this slide, big picture stuff here, is that enzymes decrease the energy of activation. And there will be a test question on that. And all that means is enzymes make it easier for the reaction to go forward because less energy is required to make the activation go. All right, we already mentioned cofactors. They can be metallic cofactors. This will be metal ions. Or they can be organic cofactors. And when they're organic, we call them coenzymes. And they're going to help in a multitude of ways, but usually they help through, bringing the active site and the substrate closer together. Sometimes they help transfer a molecule from the substrate to another substrate, things like that. All right, so let's look at this figure for a second. The purple here is the enzyme, all right. I should say the apoenzyme. This is the coenzyme, so we know it must be an organic molecule. And then this enzyme is going to be interacting with two substrates. You have substrate 1 here and substrate 2 here. And substrate 1 has a chemical group on it. So when the substrates bind to the active site, the coenzyme is going to fit right into that chemical group just so, and it's going to help transfer that chemical group from the first substrate to the second substrate. When that happens, the enzyme undergoes a shape change, and it releases the products, which now we can see substrate 1 without the chemical group and substrate 2 with the chemical group. All right, this is another chart just showing some example enzymes and their action. So we already talked about lactase being the enzyme that works on lactose, and it's going to break lactose down into its constituent parts, which is a glucose and a galactose. Penicillinase. This is produced by some bacteria, and what it does is it breaks down part of penicillin and makes it basically ineffective against the microorganism. DNA polymerase, I already mentioned, is going to catalyze the synthesis of DNA. Lactate dehydrogenase, you will see it's important in the step between glycolysis and the Krebs cycle, which we'll get to here in a little bit. And then oxidase, you'll see we'll do oxidase tests in this course in lab. And what oxidase does is it essentially adds hydrogen atoms to oxygen to form either water or hydrogen peroxide, just depending on what oxidase we're testing. All right, where are enzymes active? Well, they can be active outside of the cell, and if they are, they're called exoenzymes, or they can be active inside of the cell, and we refer to those as endoenzymes. Exoenzymes, they're going to generally break down large food particles or harmful chemicals. So examples there are going to be cellulase, which breaks down cellulose, amylase, which breaks down starch, and we'll do an amylase hydrolysis test in this course, and penicillinase, which we mentioned breaks down penicillin. And these all make sense, right? Because cellulose, starch, and penicillin are all going to be outside of the microorganism. They're going to be extracellular to the microorganism. So these enzymes are going to be active outside of the cell. Endoenzymes are honestly most enzymes, and they're going to be involved in the functioning of the cell itself. This is just showing you exoenzymes. You can see the enzymes are inactive when they are intracellular, but as soon as they're secreted, they are active and they can interact with the substrate to form products. Endoenzymes, most enzymes are endoenzymes and they are active inside of the cell. Enzymes can be constitutive, which means they're always there. They're always going to be produced at about equal rates, regardless of how much substrate is present. Regulated enzymes, on the other hand, are not always present. Their production is going to be turned on or induced, or turned off or repressed in response to whether or not substrate is present and how much substrate is present. All right, the two kind of reactions, big reactions that you can have synthesis reactions, and this is where you're building up bigger molecules from smaller molecules. These are also called condensation or dehydration reactions. I'll tell you why in a sec. Remember, anabolic reactions are going to be synthesis reactions. And these release a water molecule for every bond that's made. And you can see right here, this is forming a glycosidic bond between two glucose molecules to make maltose. So what happens is the two glucose molecules fit into the active sites on the enzyme. The enzyme changes shape a little bit, forcing these bonds closer together and is going to release a water molecule. And then the enzyme is going to change shape once more back to its original shape, and it's going to release the product, maltose. This is a synthesis reaction or an anabolic reaction because we took two smaller molecules, two glucose molecules, and built them into a bigger maltose molecule. Now, the exact opposite of that is a hydrolysis reaction, also called decomposition reactions. And we already kind of looked at one earlier when we looked at the induced fit model. These are also known as catabolic reactions because we're breaking down big molecules into smaller molecules. Because it's the opposite of synthesis, it's going to require a water molecule in order to break those bonds. Okay, here we have an example of a hydrolysis reaction. So here's our enzyme. Here's the molecule, and it's fit into the active site of the enzyme. And then it's going to take water into this area. And the enzyme is going to change shape, put pressure on those bonds, break the bonds, and a molecule of hydrogen and a hydroxide molecule are going to be added to the substrate. The enzyme is going to change shape back to its original shape, and in doing so, it's going to release the products. And what we've done here is we've taken a big molecule and we've broken it down into two smaller molecules using water in the process. So this is a catabolic reaction. I want you guys to be very familiar with the difference between anabolic and catabolic reactions. You've got to remember that anabolic is building things up. And the way I remember this is I remember anabolic steroids are used by some bodybuilders to build their bodies up. If you can remember that, you can remember the other one is the other thing. So catabolic reactions are going to break things down. Another way to think of that is cats tend to be maniacs and they tend to break things. If you own a cat, you probably know what I mean. Okay, question. So, which of the following statements about enzymes is correct? All right, so we know there's going to be 3 false statements here, so we're looking for the correct one. A, they show no substrate specificity. B, they are consumed by the reaction. C, they may be active extracellularly, or D, they are composed only of protein. Well, A, we know that's false because enzymes are highly specific for the substrate that they work on. Remember that only a very specifically shaped substrate can fit into that active site on the enzyme. B, they are consumed by the reaction. No, they are a catalyst that is not consumed by the reaction. So they speed things up, but they stick around. D, they are composed only of protein. We know this is false because some enzymes require cofactors or coenzymes that are not protein. So the answer here is C, they may be active extracellularly, and we learned about that when we talked about penicillinase. Okay, so enzymes are going to be influenced by their environment. And each enzyme has its optimum temperature, pH, and osmotic pressure that it operates at. And these tend to be the same temperature, pH, and osmotic pressure of the given organism. And that should make sense, right? For example, our enzymes tend to operate at the same temperature as body temperature. Now, when enzymes are subjected to changes in the environment, they're going to become unstable. We call chemically unstable enzymes labile. And the process that breaks the bonds of the enzyme and deranges its shape is called denaturation. Things like high temperature, acidic pH, these things can disrupt those bonds of the enzyme, of the protein, and make it take on a completely different shape. And when it does that, it loses its function. Remember, the shape of an enzyme is what makes it able to function. It's what makes it able to bind to a specific substrate, is because that substrate and the enzyme's active site are complementary in such a way that they fit together like a lock and key. If you mess with the structure or the shape of that active site, you're going to mess with the ability of the enzyme to do its job, to act on the substrate. And when those bonds are broken, again, we call that denaturation. An example of this would be when you fry an egg. The reason that the egg whites turn white is because you're looking at that protein, albumin, become denatured. So why do we talk about enzymes when we talk about metabolism? Well, you'll see that enzymes are integral to metabolic pathways. And a metabolic pathway is just a stepwise, highly regulated pathway in which the cell uses available nutrients and energy to its maximum use. Metabolic reactions are going to be chosen by the cell to favor its growth and survival in specific environmental conditions. And what that means is just that the organism evolves in order to have precise metabolic reactions that make sense given the organism's environment, okay? For example, it would not make sense for an organism to have pathways to break down glucose if the organism lives in an environment without glucose. That would just be a waste of the organism's energy. And you'll see, in order for the organism to optimize its energy, it has evolved checks and balances that regulate the enzymes. And we'll talk a little bit about that in a moment. Metabolic pathways can take on several different patterns. You can see there are going to be multi-enzyme systems here. So we have a cyclic system where you've got a reactant that gets turned into a product that then becomes a part of a cycle. And each one of these arrows represents an enzyme requiring step. Here's a linear pathway. This is very easy to follow, and we'll talk about linear pathways, or we'll see a linear pathway when we go over glycolysis in a moment. And then the pathways can diverge, they can converge. It just depends on the needs of the organism. So how are the actions of enzymes controlled? Well, one way to control enzyme is to inhibit it. There's competitive inhibition and non-competitive inhibition. Now, competitive inhibition is going to involve a substance that looks like the normal substrate. So it's going to have a shape similar to the normal substrate, and it can therefore compete for binding to the active site. All right, so it's competing against the substrate to make contact with that active site. Non-competitive inhibition, on the other hand, is not going to involve the substance binding to the active site. The one I want you to be familiar with is allosteric inhibition. And this is where an enzyme has two binding sites, the active site and then the regulatory or the allosteric site. And what happens is the inhibitor, the substance, comes in and binds to the allosteric site, and that causes the enzyme to change shape, and it can no longer bind substrate at the active site when it changes its shape. You'll see here in just a second that enzymes are going to be regulated by negative feedback from the binding of molecules, usually the products of the reaction at this allosteric site. So on the left here, we have an example of competitive inhibition. Here's your enzyme. Here's its active site. Notice the unique shape. Here's the normal substrate. Look at the shape of this substrate. You can see how it can fit into the active site. But so can the competitive inhibitor. So both molecules are competing for the active site. If the substrate binds to the enzyme, then the reaction is going to proceed normally. However, if the competitive inhibitor binds to the active site, the substrate is going to be blocked from binding, and you'll notice that the competitive inhibitor does not turn into the products that this enzyme catalyzes. So the reaction is going to be blocked. Now, on the other side here is allosteric inhibition. You can see the enzyme, you can see the active site, and then here's the regulatory site. Here's the substrate. So the substrate binds to the active site. It produces products, but one of these products comes around and binds to the regulatory site on the enzyme. And that closes this active site off, and no more substrate can bind, thus stopping that reaction. And you'll see this is a very common tool used in metabolic pathways, where the product goes back and inhibits the enzyme of the product's reaction that it was just involved in. And that's negative feedback. A lot of medications are competitive inhibitors, and I want to show you aspirin as an example. So this is the pathway for prostaglandin synthesis. So you've got a stimulus occurs, the cell takes some membrane phospholipids from that cell membrane, gets arachidonic acid from those phospholipids, and then it's going to take arachidonic acid, and depending on the stimulus, it's going to turn it into certain prostaglandins. Under normal physiologic conditions, this enzyme, COX-1, cyclooxygenase 1, is the pathway of prostaglandin synthesis. And these prostaglandins that get made help with GI mucosal integrity, so they help protect the lining of your stomach. They help your platelets function, and they help your kidneys function. So these overall are pretty good prostaglandins. However, in the presence of some sort of inflammatory stimulus, let's say arthritis, you're going to have the arachidonic acid go down the COX-2 pathway. And the COX-2 pathway makes these prostaglandins here. So you may notice that these products here, these prostaglandins, are the same prostaglandins over here. So what's the difference? Well, COX-2 is only turned on or induced, and we'll talk about that in a second, during inflammatory conditions. So normally this enzyme isn't present when there's inflammation going on. COX-2 is going to be made by the cell, and the COX-2 pathway is going to be utilized. So what happens is you get like an increase in prostaglandin levels overall in the body. What aspirin does is it's a non-selective COX inhibitor. That means it's going to come in and it's going to block COX-2. So it's going to reduce the amount of prostaglandins, right, and reduce the overall inflammation the body's experiencing. But unfortunately, it also blocks COX-1. So we don't get these prostaglandins that are beneficial to the stomach lining, platelet function, and kidney function. So that's why aspirin can have negative side effects for your stomach. It can cause bleeding problems, and it can also decrease kidney function. So aspirin is a competitive inhibitor of both COX-1 and COX-2. And what that means, as you now know, is that aspirin competes to bind at that active site on both of those enzymes. And so it's competing with arachidonic acid for binding on those enzymes. Another way to regulate enzymes is to regulate them at the genetic level. So you can have repression or induction of an enzyme. And what that means is essentially if you repress an enzyme, you're turning off the gene that produces that enzyme. And then if you induce an enzyme, you're turning on the gene that produces that enzyme. And I mentioned COX-2 is induced during inflammatory conditions. For example, let's say a staph infection. So the skin is going to become red and inflamed. And in those cells, the COX-2 gene is going to be induced. So those cells are going to produce more COX-2 enzyme. And that COX-2 enzyme is going to catalyze the conversion of arachidonic acid to prostaglandins, increasing the amount of prostaglandins in the area and causing things like pain, swelling, things like that. So again, repressing an enzyme or repressing the enzyme's gene, you're going to turn off reading of that gene. So the product does not get made, the enzyme does not get made. And induction is going to be the exact opposite. You're turning on the reading of that gene so that more enzyme can be produced. How do we repress a gene? Well, here we have the strand of DNA. This strand of DNA is going to get copied over into RNA. Remember, this is the process of transcription. So we have an RNA instructional copy of a gene, and we're going to turn that, this is the gene for an enzyme, so now we need to turn it into a protein, and this is translation. Now that we've got this strand of protein, remember this protein is going to fold in on itself until it takes on its functional form. And then you've got your enzyme. Now the enzyme and the substrate react together to form products. And what happens here is the products of that reaction go back to the DNA and repress reading of that gene. Okay? So they turn off that gene. So there's no RNA copy made, or transcript made. And then there's no translation that occurs, so you can't make the protein. So overall, you get no enzyme made when you repress the gene. All right, energy. We're going to talk a lot about energy, what energy is, what it's used for, and then how we derive energy in the cell. Energy can take on several different forms, so thermal, radiant, electrical, mechanical, atomic, and chemical energy. Cell energetics here. So cells are going to manage their energy through chemical reactions that make or break bonds, and in doing so, transfer electrons. All right, first of all, I want you to know that chemical reactions are reactions that make or break bonds. That's going to be a test question. Now, when these bonds are made or when these bonds are broken, electrons are going to be transferred. You have two basic kinds of energy reactions. You have endergonic reactions, which consume energy overall. So you got A plus B, but they require energy and an enzyme to become C. Or you can have exergonic reactions. These release energy overall. So you have X and Y plus the enzyme goes to Z plus energy that's released. Now, that released energy is typically temporarily stored in a high energy phosphate molecule that we call ATP. The cell then uses that ATP for its endergonic cell reactions. All right, when we talk about the transfer of electrons during chemical reactions, we have to talk about oxidation reduction reactions. These are also called redox reactions, okay? These are always going to occur in pairs. One member of the reaction is going to be oxidized while the other member is simultaneously reduced. One member is an electron donor. The other member is an electron acceptor. Okay? Now, the member that gets reduced gains electrons, and the member that gets oxidized loses its electrons, some of its electrons. How do you remember this? Well, the way they named this is when they say reduce, they're talking about reducing the molecule's overall charge. So let's say you have a molecule that has a +1 charge. It undergoes a redox reaction, and now it's neutral. Has it lost or gained electrons? Well, the charge went from +1 to neutral, so we know it gained a -1 charge. It gained an electron. And its overall charge has been reduced from +1 to 0, right, to neutral. That's the way you can keep this in your head, is remember that reducing is talking about reducing the charge of the molecule. This process overall is going to salvage electrons and their energy, and then that energy can be captured into ATP or another compound. All right, so there's a lot going on this slide. I'm going to try to take it step by step and simplify it. But this is just to kind of introduce you to redox reactions and what their purpose is. It's also to introduce you to the metabolic pathways we're going to be talking about here shortly, which are glycolysis, the Krebs cycle, and oxidative phosphorylation. All of these together make cellular respiration or aerobic respiration, okay? And this is how the cell makes its energy, is using these pathways. Now, the thing I want to show you is along these pathways, we have a lot of reduction of molecules. So this molecule here, NAD, you see it comes together with glucose, and there's an enzyme here that's working as well. And it produces a molecule of pyruvate, and it produces a molecule of NADH. Now, NAD went from having a positive charge to having a neutral charge. So it's been reduced. It gained electrons in this reaction. Glucose, therefore, must have lost electrons during the reaction. So glucose becomes oxidized to pyruvate. Here is pyruvate being oxidized to acetyl-CoA, and you'll see the same exact thing here. NAD becomes reduced to NADH. What's the point of this? Well, the point is that when you take these electrons away from organic molecules like glucose, nutrient molecules, you are transferring energy temporarily to these molecules. And these molecules take that energy in the form of electrons, and they take it to the electron transport system. And what the electron transport system does is it takes those electrons and adds it to oxygen. And in doing so, it produces ATP from ADP. So you'll see redox reactions are incredibly important when it comes to utilizing nutrients that the cells take in. So we're also going to talk about all of these pathways. I just want to give kind of a brief overview. Glycolysis is going to be the breakdown of glucose into pyruvate, and we'll talk about it in detail. There's an intermediate step here we won't really talk about, but just know that pyruvate goes on to become acetyl-CoA. Acetyl-CoA participates in the Krebs cycle. And what I want you to know about the Krebs cycle is that redox reactions occur there. So you're going to be getting energy from glycolysis, right? You're going to be getting energy from, or potential energy, electrons from the intermediate step. You're going to get electrons from the Krebs cycle. And all of these electrons go onto the electron transport system, also called the electron transport chain, where they get transferred over to oxygen. Oxygen is the final electron acceptor. And in doing that, the cell is able to make ATP, and I'll show you how here shortly. Okay, so we said electrons are going to be highly reactive. We know they have a charge of -1, and they are transferred or shared between atoms during bond formation, during chemical reactions. In metabolism, electrons are going to be captured, and they're going to be transported by carrier molecules throughout aerobic respiration. That's what I just talked about, okay? Electrons are then going to be transferred to oxygen, the final electron acceptor. I want you guys to know that. The final electron acceptor in aerobic respiration is oxygen. In doing that, what happens is a proton gradient is built. A proton is just a hydrogen ion, right? Hydrogen atom consisting of one proton and one electron. You take that electron away, all you're left with is a proton. So, as these electrons are being shuttled through the electron transport system, what happens is a proton gradient builds up. And it's that proton gradient that actually activates the enzyme that produces ATP. Why do we produce ATP? Well, ATP has high energy phosphate bonds, so that energy gets stored in those phosphate bonds. All right, electron carriers. So remember, these electrons are being shuttled. They're being taken from glycolysis. They're being taken from the Krebs cycle, and they're being moved over to the electron transport chain, which is itself a chain of enzymes and electron carriers that move those electrons sequentially through the chain until they reach molecular oxygen. Most of these carriers that are involved are going to be coenzymes. So NAD and FAD, coenzyme A, coenzyme Q. There's a lot of coenzymes involved. Just know that essentially organic molecules are taking these electrons and transferring them to oxygen. Okay, question. If a molecule has been reduced during a reaction, it has... Well, remember, reduced is referring to its charge. So I always think of the example, a molecule with a +1 charge, so an ion with +1. If its charge is reduced to a 0, that means it had to have gained a -1 charge. Therefore, it had to have gained an electron. So the answer here is D. Remember, the reduced agent is always going to be the agent that gained electrons. Oxidized agent loses electrons. Okay, so why are we so obsessed with making ATP in the cell? Well, ATP is going to be the usable form of energy that the cell has. It's essentially like currency. ATP is a 3-part molecule. It has adenine, which is a nitrogenous base. You find it in DNA and RNA. It has ribose, which is a 5-carbon sugar. You find that in RNA. And it has 3 phosphate groups, right? Adenosine triphosphate. So 3 phosphate groups, and it's those phosphate bonds that hold energy. In fact, it's the final phosphate. If you take that off of the molecule, it's going to release energy. And the cell is going to use ATP for its reactions that require energy. This is showing you ATP, right, with the 3 phosphates on it. The molecule that just has 2 phosphates is ADP, adenosine diphosphate. And then when it has just 1 phosphate, it's adenosine monophosphate, okay? These are the bonds that have energy in them. The terminal bond, the bond between the terminal phosphate being the most energetic. Okay, so ways that the cell makes energy depends on the organism, right? So a lot of cells undergo respiration. Some undergo fermentation. Then your photosynthetic organisms are going to undergo photosynthesis, and then you also have chemoautotrophy in some organisms. And all of these processes are going to convert ADP into ATP. And that ATP goes on to do chemical work, transport work, and mechanical work for the organism. All right, as far as making ATP, there's essentially 3 different mechanisms. The first is you can have a molecule come along that has a phosphate group sticking off of it, and it just transfers that phosphate group to ADP, forming ATP. This is called substrate level phosphorylation. And so this is going to directly transfer phosphate to ADP. The other mechanism or the other strategy is oxidative phosphorylation, and we'll talk about this in a little bit. But this is where an organism uses a series of redox reactions, so a way to transfer electrons down a respiratory pathway, such as the electron transport chain. And then there's photophosphorylation, and this you'll see in photosynthetic organisms. All right, the pathways of bioenergetics. So what's bioenergetics? It's the study of the mechanisms of cellular energy release. This is going to include your catabolic and your anabolic reactions. What we're going to talk about is the catabolism of fuels, primarily glucose, and the coupled pathways in which glucose breakdown proceeds. So we'll talk about glycolysis. We'll talk about the Krebs cycle, and then at the end we'll talk about the respiratory chain where electron transport and oxidative phosphorylation takes place. So what are the strategies for metabolizing nutrients? Well, it varies, but generally it's going to be based on 3 catabolic pathways, and these pathways convert glucose to carbon dioxide and produce energy. So we have aerobic respiration. You'll see this in aerobic organisms such as us, where you undergo glycolysis, the Krebs cycle, and a respiratory chain where oxygen is the final electron acceptor. You can have anaerobic respiration. This is going to be glycolysis, Krebs cycle, and a respiratory chain as well. But at the end of this respiratory chain, oxygen is not the final electron acceptor. And then finally, you can have fermentation. This is a low energy producing process in which only glycolysis is involved. Okay, so here are those three catabolic pathways we just talked about. Here's aerobic respiration. So we've got glycolysis, which produces 2-pyruvate from a glucose molecule, and it's going to give off a little bit of energy, and it's going to shuttle electrons to NADH. Okay. Next, you have the intermediate step. Pyruvate gets turned into acetyl-CoA. Carbon dioxide is given off there, and you've got electrons going to NADH again. Next, we enter the Krebs cycle, and what I want you to see here, the Krebs cycle is going to give off carbon dioxide. It's going to produce some ATP, and it's going to transfer electrons to more molecules that go on to carry those electrons to the electron transport system. These electrons get shuttled down the series of enzymes and coenzymes until they are transferred to oxygen. Oxygen is the final electron acceptor in aerobic respiration. You need to know that. The total ATP that's possible with aerobic respiration is going to be 38 per glucose molecule. Next, you have anaerobic respiration. So you're going to see glycolysis, the intermediate step, the Krebs cycle. You're going to see the electrons being transferred to these molecules, which then take the electrons to the electron transport system. The electrons are going to be shuttled down the enzymes and the coenzymes until they reach a non-oxygen electron acceptor. So sulfate, carbonate, nitrate, etc. The ATP with this is going to vary just depending on what the electron acceptor is and things like that. Finally, you have fermentation, and you'll notice here is just glycolysis involved. Then you're left with pyruvate, which can undergo fermentation depending on the organism to either become lactic acid or some other kind of acid, or to become alcohol and carbon dioxide, or some other kind of alcohol and gas. Maximum ATP with fermentation per glucose molecule is just 2. Okay, aerobic respiration. Remember, it's going to be these 3 coupled pathways. Glycolysis, the Krebs cycle, also called the tricyclic acid cycle, also called the citric acid cycle, and the electron transport chain or electron transport system. We said these are going to be enzyme catalyzed reactions. So these metabolic pathways involve a lot of enzymes. And the big picture here is that electrons are going to be taken from a nutrient and are going to be transferred to oxygen. And in doing that, ATP is produced. So you have glycolysis. Big picture here, glucose is split into 2-pyruvate or pyruvic acids. And then you're going to generate a little bit of ATP and NADH, right? And where does NADH go? Remember, it's going to take those electrons to the electron transport chain. Next, you have the Krebs cycle. Big picture from the Krebs cycle, some CO2 is made. And again, these guys that carry electrons to the electron transport chain. And then finally, you have the electron transport chain. This is just a series of enzymes and coenzymes that catalyze sequential redox reactions. And this series of redox reactions is called oxidative phosphorylation. What happens at the end of this process is a lot of ATP gets made. Okay, so here's glycolysis. You take a glucose molecule, it has 6 carbons, and it undergoes a series of enzyme catalyzed reactions. You'll see it requires a little bit of ATP input at the beginning of glycolysis. but then it's going to produce twice as much ATP towards the end of glycolysis. So your net ATP production is 2. Pyruvate is a 3-carbon molecule down here. So you can see glucose, 6-carbon molecule, gets split into 2 pyruvates, 2 3-carbon molecules. Okay, so what happens to pyruvate? Well, you already know what happens during the respiration pathway. Pyruvate is going to go on to be broken down into acetyl-CoA, which goes on to participate in the Krebs cycle, and this generates a lot of those electron-carrying molecules that take the electrons onto the electron transport chain. However, pyruvate can also go on to be used in the synthesis of amino acids, sugars, and fat metabolites, or in organisms that undergo fermentation. Pyruvate can go on to become certain acids and gas, or even alcohol, depending on the organism. Okay, so this is the Krebs cycle. It looks crazy complicated. What I want you to take away from this is the molecules that I'm pointing at here. All of these guys are reduced forms of NAD. So what NADH has is it has electrons that it got from metabolizing that pyruvate, right? Metabolizing those nutrients. And it takes those electrons to the electron transport chain. And you can see, so we've got, here's the intermediate step, pyruvate to acetyl-CoA. So we get 1 NADH there. And then here's the Krebs cycle. We get an NADH here, here. We get an FADH2 here. This is another guy that carries electrons. And here's another NADH. So all of these molecules are carrying electrons to the electron transport chain, where energy is going to be made using that energy from the electrons. All right, electron transport and oxidative phosphorylation. We said this is going to be where the final processing of electrons occurs. This is also where most of the ATP is made. You need to know that. What it is it's a chain of redox carriers that are going to receive electrons from the reduced carriers, NADH and FADH2. The electron transport system shuttles those electrons down its chain. And remember, I said a protein gradient is generated. And it's that protein gradient that fuels or activates ATP synthase. And ATP synthase goes on to produce ATP. So where does all of this occur? Well, in eukaryotic organisms, it's going to take place in the mitochondria. This is why the mitochondria are called the powerhouses of the cell. All the ATP, essentially all the ATP, is made there. Now remember that a mitochondrion has 2 membranes. It has an outer membrane, this smooth membrane here, and then it has this convoluted, highly folded inner membrane. And those folds are called cristae. And on each fold are the enzymes and the coenzymes involved in shuttling the electrons towards oxygen. You can see ATP synthase is also there. This is the enzyme that helps catalyze the production of ATP. So what happens is those molecules that we've talked about, NADH and FADH2, from glycolysis in the Krebs cycle, are going to come into the cristae, and they're going to interact with these enzymes, and they're going to transfer their electrons to the enzymes. Now, let me point something out here. Electrons are not going to be transported by themselves. They're actually transported in the form of a hydrogen atom. Remember, a hydrogen atom is 1 hydrogen ion, or a proton, plus 1 electron. Now, when these molecules get to this point of meeting the enzymes, what essentially happens is that hydrogen atom gets split into a proton, which gets pumped across the inner membrane into this space here, and an electron, which gets placed on the electron carriers of the chain. Okay, so the electrons are moving down the chain towards oxygen here, while the protons are getting pumped across the inner membrane into the inner membrane space. Okay, this is the inner membrane space here. The inner part here is the matrix of the mitochondria. Now, why is this proton gradient important? Well, you'll notice there's a high concentration of protons in the inner membrane space and a low concentration in the matrix. What happens is this gradient, these protons move through ATP synthase, and it's essentially activate this enzyme to help phosphorylate ADP, producing ATP. So you have to have this proton gradient in order to produce ATP. That proton gradient is also known as chemiosmosis, because if we go back, not only is there a concentration gradient established, so more protons on this side than on this side, but there's also a difference in charge. So it's more positive on this side than on this side. Both those things drive the protons across ATP synthase into the matrix, and that's known as the proton motive force. Now, when those hydrogen ions or protons diffuse back, they go through the ATP synthase, and this actually causes the molecule to rotate, resulting in a 3D change of shape, and that 3D change of shape is what catalyzes the production of ATP. That is cellular respiration. Now, that's all great in eukaryotes, but what about in prokaryotes? Prokaryotes lack mitochondria, so how can they make their ATP? Here you can see a close-up of the bacterial cell envelope. So we have the cell membrane, we have the cell wall. This is the periplasmic space, remember, the space between the cell membrane and the cell wall. And then here's the electron transport chain. And here's ATP synthase. The bacterium essentially does the exact same thing. It just, instead of using mitochondria, it uses its own cell membrane. It sets up the chemiosmotic gradient here. And then these protons move through ATP synthase, and that catalyzes the conversion of ADP to ATP. Okay, in aerobic respiration, remember, oxygen is the final electron acceptor. The electron transport chain essentially stops with oxygen. Those electrons are transferred over, and the proton gradient has been established, and that proton gradient fuels the synthesis of ATP. Your net result of aerobic respiration, you can see you take some glucose and you take some water, oxygen, and then you take some ADP and some inorganic phosphate, and you turn that into CO2, water, and a bunch of energy. Notice here, CO2 is produced. This is a waste product. The cell has to get rid of this CO2, or I should say the organism has to get rid of this CO2. This is why we breathe off CO2, is that it's made at the cellular level during aerobic respiration. And oxygen is the final electron acceptor, and that's why we have to bring oxygen in. All right, this is showing the theoretical yield of ATP during each step of aerobic respiration. So you can see here during glycolysis, only 2 ATPs are made. or the net output is 2 ATPs. Compare that to oxidative phosphorylation, you've got 34 ATPs. That's kind of the big takeaway that I want you to know is glycolysis. If you are an organism that can only undergo glycolysis, like a fermenter, your ATP output from glucose is going to be low. You're only going to get 2 ATP per glucose molecule. Whereas in an organism that can use oxidative phosphorylation, you're going to get something more like 38 ATP per glucose molecule. All right, question here. What part of aerobic respiration releases CO2? So go back and think. So glycolysis, what is this going to produce? It's going to produce 2-pyruvate and a net 2 of ATP. Chemiosmosis is not part of the pathway. The electron transport system. Now remember, that's going to set up a proton gradient and it's going to deliver electrons to oxygen. So the answer here is going to be B, the Krebs cycle. What's happening there is pyruvate gets broken down into acetyl-CoA. Acetyl-CoA combines with another molecule, forming a 6-carbon structure, and those carbons are sequentially removed during the Krebs cycle to produce CO2 and to transfer electrons over to those electron-carrying molecules, NADH and FADH2. So the answer here is the Krebs cycle. All right, anaerobic respiration, the things I want you to know about anaerobic respiration. It's not going to use oxygen as the final electron acceptor. It's going to use some sort of oxygen-containing ion, like nitrate or nitrite. Overall, anaerobes are going to produce much less ATP than aerobes do, okay? And again, that's because they lack the ability to utilize an aerobic electron transport chain. They may use an electron transport chain, but they're going to be reducing some sort of compound other than oxygen. All right, and here it is, just showing you anaerobic respiration. You have glycolysis, you have the Krebs cycle, you have electron transport. And remember, these electrons are going to get shuttled down the electron transport system, and they're going to go to a non-oxygen electron acceptor. Okay, a little bit about fermentation. This is going to be the incomplete oxidation of a nutrient molecule, such as glucose, without the use of oxygen. Fermentation is essentially only going to go through the glycolysis step. And we will do some biochemical tests on organisms later on in this course. And one of the tests we'll do is see if they ferment glucose. Fermentation is going to yield a very small amount of ATP. Some yeasts acting on glucose, some microorganisms that act on glucose can produce alcohol as a byproduct. And this is essentially how we brew beer. Other organisms, again, depends on the organism, can form acid, gas, and other byproducts. And this is from the action of the bacteria on pyruvate. And we will do some biochemical tests in this course, and you'll be looking for evidence of acid production, gas production, etc., depending on the bacteria we're studying. Okay, so here's fermentation. You can see we have glucose, and it only undergoes the glycolysis step. It ends up in 2 pyruvate molecules, and that pyruvate goes on to either form an acid or an alcohol. Okay, here it is a little bit more in detail. So let me just point out this chart to you because it's a little confusing to look at first. On the left, we're going to have a reaction that we're going to talk about that happens in yeasts. On the right, we're going to have a reaction that happens in human muscle. Here we have a glucose molecule, and it gets converted into pyruvate. Now, what happens to the pyruvate depends on the organism. In our muscles, pyruvate goes on to form lactic acid, and in doing this, it takes electrons from NADH, okay? So it oxidizes NADH into NAD. That's in human muscle. Now on the left, we have the organism of yeasts. So what yeasts do is they take pyruvate, they form acetaldehyde out of the pyruvate, and then they form alcohol. And the way they do this is they take electrons from NADH, they oxidize NADH to NAD. Now, what's the purpose of this? The big picture is the organism has to find a way to regenerate NAD, or else glycolysis and everything that's after it is going to shut down. And when that happens, the organism can't utilize energy, it can't break down nutrients, and it's going to die. Okay, big takeaways here. Aerobic respiration. I want you to know this is going to consist of glycolysis, the Krebs cycle, and electron transport, and you have to know the final electron acceptor in aerobic respiration is oxygen. You'll see that it's going to be found in aerobes and facultative anaerobes, these pathways, okay? So these organisms have the ability to utilize these pathways. Anaerobic metabolism, on the other hand, is going to be fermentation. This is only going to involve glycolysis. You'll see this in strict anaerobes. These guys lack those enzymes. They lack the ability to produce these pathways of the Krebs cycle and electron transport, okay? And they do this glycolysis in the absence of oxygen. And then you have just respiration. Remember, this is going to involve all the pathways, but there's going to be an inorganic ion. That's the final electron acceptor. All right, the crossing pathways of metabolism. Just zoom out for a minute and realize that most of these pathways are going to have multiple directions that they flow in. This is because those molecular intermediates that are produced along those pathways can be used in other pathways, and it really just depends on what the cell needs. For example, pyruvate, we said, can be converted into amino acids. That's through a process called amination. Those amino acids can be converted into energy sources. And then you've got, like, for example, glyceraldehyde 3-phosphate. This guy's from glycolysis. It can go through various pathways to become amino acids, carbohydrates, or fats. So just know there's a lot of different metabolic pathways. They kind of flow together. They come in at different points. Different byproducts can be used in different pathways. And it's all based on what the cell needs at any given time. Okay, photosynthesis, just a note here. Big picture, the ultimate source of all chemical energy in cells is going to come from the sun. And the reason for this is, of course, autotrophs. Remember, autotrophs are going to produce their own energy. They're going to produce organic molecules, nutrient molecules, using water, CO2, and energy from the sun. And heterotrophs feed on autotrophs, as well as other heterotrophs. It just depends on the organism. But ultimately, you can trace back all energy to the sun. Now, on land, we tend to think of plants as being the primary photosynthesizers, which they are. But in aquatic ecosystems, the photosynthesizers are going to be algae, cyanobacteria, and then some sulfur-producing and non-sulfur-producing bacteria. And realize that the aquatic ecosystem organisms are going to account for 80 to 90% of all photosynthesis that occurs on Earth. So while green plants are the primary photosynthesizers on land, the main photosynthesizers in general are your aquatic guys. How does photosynthesis occur? It's going to occur in two stages. You have light-dependent reactions. Photons are going to be absorbed by those pigments like chlorophyll. This is going to split water, releasing oxygen. And then that's where the organism gets its electrons from to drive photophosphorylation. And in photophosphorylation, ATP is synthesized. Then you've got light-independent reactions. And just know the organism uses ATP to fix CO2, so basically to take CO2 and ultimately convert it to glucose. So that's how the organism, the photosynthetic organism, makes its nutrient molecules. All right, question. The main job of chloroplast pigments during photosynthesis is, and then if you reach back pretty far and remember that chloroplast pigments, such as chlorophyll, are going to capture photons of light, making that light usable during photosynthesis.